Say hi, Ruben.
Oh,
Hi, guys. We're continuing with my notes for the life science in a nutshell. So we're reading the review, and okay. So anyway, we are here learning about the cytoskeleton of cells, and which are consist of microtubules, microfilaments made of actin and are actin filaments and intermediate filaments, and these interact with proteins called septins and spectrin. So we learned about the different kinds of components of the cytoskeleton, and so yeah, this is inside the nucleus. This is showing intermediate filaments. This is microtubules. This is microfilaments. This is the muscle cell, the green on the bottom. This one is the cytoskeleton, showing microtubules in green and active filaments in red. A very nice picture. So we have these proteins that interact with the cytoskeletal filaments, and there's a lot of there's a lot of different proteins that interact, but major proteins called septins, which we reviewed in the last video, shown here in the green stain, and around the peripheral of cells and replicating cells, etc. They help with the control of the shape, the cells budding, and in the separating of the cells and the mitotic spindle and cytokinesis and etc. And they interact with the membrane of the cell. And now we're learning about a second kind of protein which also helps with the shape changing of the cytoskeleton. This is called spectrin. Here in the picture it's shown around cells. It's in the green color, and the blue is the inside of the nucleus of the cell. As you can see, the nucleus is a big part of the cell. Okay, so this is showing a subunit diagram of how the spectrin interacts with actin, etc. Okay, go ahead and read this. Spectrin forms.
spectrum forms pentagonal and hexagonal arrangements, forming a scaffolding and playing an important role in maintenance of plasma membrane integrity and cytoskeletal structure. The hexagonal arrangements are formed by tetramers, spectrin subunits associating with short actin filaments at either end of the tetramer. These short actin filaments act as junctional complexes, allowing the information of.
Okay, start over.
These short actin filaments act as junctional complexes, allowing the formation of the hexagonal mesh.
In the light microscope, the basic shape of the red blood cell could still be seen as spectrin containing submembranous cytoskeleton preserved the shape of the cell in outline. So these spectrins are found in red blood cells, also called erythrocytes. So the way the spectre, meaning a ghost, so the spectre or stays, or the outline of the cell stays, um, reflecting what the cytoskeleton looks like of the cell. And that's how the names came to be for spectrum, which is the protein interacting with the cytoskeleton. So, go ahead. Suki.
Association with the intracellular phase of the plasma membrane is by indirect interaction through direct interactions with cytoskeletal
No.
preserve the shape.
No, it's protein and ankyrin.
Protein and
Interactions with protein
accurate
and ankyrin with the transmembrane ion transporter bound 3 protein, binding the spectrum tail region to the transmembrane protein glycophorin A. In animal, spectrum forms the meshwork that provides red blood cells erythrocytes their shape, so it interacts with other proteins as well. On the membrane, etc. Dimeric spectrum, meaning two units, is formed by the lateral association of alpha and beta monomers to form a dimer. Dimers associate in a head-to-head formation, producing the tetramer. So tetra means 4. I believe 4. Is it? Or 5 cards. End-to-end association of these tetramers with short-actin filaments produces the hexagonal complexes. So I double checked, and in fact tetramer means 4 monomers or subunits. So the spectrum gene family has undergone expansion during evolution. Rather than 1 alpha and 2 beta genes in invertebrates, there are 2 alpha spectrums and 5 beta spectrums named in order of discovery. So in humans, the genes are alpha, beta, and whatever. SPTA1, 2, on and on till 5. There's a lot of them. Production of spectrum is promoted by transcription factor GATA1. In myocardial cells, all spectrum distribution is coincident with Z-discs and plasma membrane of myofibrils. It is important in sarcomere morphology and calcium homeostasis in the myocardia. So, just to note that sarcomere, so myofibers are also referring to muscle fibers, also found in the heart, like cardiac muscle, different kind muscle. And a sarcomere is the basic unit of the muscles. It's a muscle cell, basically. It's the basic contractile unit for striated and cardiac muscles, made up of parallel actin, which are thin filaments, and myosin, or thick filaments. Interaction of myosin and actin proteins causes a contraction of the muscle cells. So let's see. So myocardia is referring to the muscles in the middle layer of the heart. So it's cardiac muscle. And myofibrils are just muscle tissues. So again, it consists of actin proteins, myosin, and a giant protein called titin. So skeletal muscles are composed of tubular muscle cells, myocytes called myofibers or muscle fibers also, and they're formed in a process known as myogenesis. So it says the cytoskeleton was once thought to be a feature only of eukaryotic cells, but homologous to all the major proteins of the eukaryote cytoskeleton have also been found in prokaryotes. So in the next section, it's comparing eukaryotes and prokaryotes briefly to see how similar they are in their cytoskeleton.